So now we're on key area 6 on the first unit, and that is we're going to be looking at mutations. What is a mutation? Well, it's just basically a change in the structure or the amount of an organism's DNA. Now, mutations arise spontaneously and at random. It's pretty rare, a mutation. In humans, it's estimated there's about 1 mutation per 15 to 30 million base pairs. That's not something you need to know for your higher exam, but it's just to give you some indication of quite how rare it is. But mutation rates differ in different genes and in different organisms. There are some genes that are more prone to mutations than others, and some organisms that are more prone to mutations than others. So we're going to have a look in this video and in the second video that I'm going to put together for this at the types of mutations. In this video we're going to look at these two types of mutations here, a single gene mutation, and then we're going to look at what is called a splice site mutation. In the following video we're going to look at chromosome mutations, and we're going to look at something called polyploidy. On single gene mutation. What is a single gene mutation? Well, it's involving a change in one of the base pairs in the DNA sequence of a single gene. Sometimes they're also known as point mutations, and they might occur in the protein coding sequence or in the regulatory sequence, which control the expression of a gene. Let's have a look at the examples of the types of single gene mutations that you can get. Well, I've put this up here called digs to help you remind you of the names of these. There is one called deletion, a type called insertion, and a type called substitution. Now, some of these names are a wee bit similar when we come on to chromosome mutations, so that's why I've put together this digs to help you remember that deletion, insertion and substitution are found in gene mutations. And you'll see the reason why I've done this when you come on to the chromosome mutation. So look at deletion mutation. So a deletion mutation is really where you lose a single nucleotide on the piece of the DNA where the gene is. So if we could imagine here that this is a gene, and here is ultimately the protein that's going to be made from that gene here. If we have a deletion of a single nucleotide, and in this case this A here ends up being deleted, then what happens is that following that deletion, every single amino acid along the protein chain has been changed, and that may result in the production of a malfunctioning protein. You can have an insertion mutation. So again, here's the gene, but this time here what has happened is that between the T and the C up here, we've had an A inserted, and that results in different amino acid order than you would have had up here, and that may produce a malfunctioning protein as well. Now, insertion and substitution are both also known, or have this description here, called frame shift mutations. And that's because the deletion or the insertion, after that deletion or insertion, the next triplet code is shifted. It's either shifted forward or it's shifted backward. And that means that every single amino acid after the insertion or the deletion has been changed. And so therefore it's more likely that it's generally more harmful, since all the amino acids in the protein will have been changed from that mutation onwards. So when you see this term frame shift mutation, you have to think of the fact that everything has either been shoved along. All of these bases that are here are either moved forward or moved back a base, depending on whether it's an insertion or a deletion. And that's why we call it a frame shift mutation. The third mutation that we have is this one here, is called substitution. And what they mean by the substitution is that you don't lose or gain a base, but what happens is, you'll see here, this amino acid, this base here, adenine, has been replaced by cytosine. Now what happens with that is that only that amino acid there has been changed. And so that may or it may not produce a malfunctioning protein. And there are different sort of types of substitution, and we'll go on to have a look at those different types. So these substitutions are not frame shift mutations, but they fall into these categories. I've got 5 here, but I only want to say 4. These 4 that are here. Silent mutations, neutral mutations, missense mutations, and nonsense mutations. So we'll go on to have a wee look at these. So a silent mutation. In this case, you've got a nucleotide that's substituted, and the new triplet code, it codes for the same amino acid. So say you've got C, C, T, and it ends up that the C is replaced by an A, but by some chance it happens that it's the same amino acid that this codes for, then it won't really affect the final protein, so the protein is therefore normal. That's your silent mutation. It's happened, but it's not really noticed in the final protein. The next one is the neutral mutation. And what happens there is that you've got the substituted nucleotide, so you've got this, but so the nucleotide here is substituted for an A, say, there. But what happens is you do get a different amino acid, but it's very similar to the original one. And that means that the protein that's been formed is very, very close to the original. And it means that, and the mutated protein will still function properly. So it's known as a neutral mutation. And we're kind of moving along these here to the ones that will actually be a bit more serious. So here is the missense mutation. And what results with that is the substituted nucleotide will code for a different amino acid, but unlike the neutral one that was there that we've looked at just a moment ago, this time round the amino acid that is substituted there, it changes the function of the protein, and that could have quite a serious effect. So you have a malfunctioning protein there. The last one is this nonsense substitution. So what happens here is that the nucleotide or the base that is substituted results in a stop codon being formed. And so that means when the stop codon is there, it means that the protein chain that you were trying to form, or the polypeptide chain, is cut shorter because it's stopped. And you may get a functioning protein depending on where that stop has happened. If it's pretty near the end, you might get away with it, but if it's right in the middle, you will get a different protein that is made, and so therefore it won't work the way that it's supposed to. I've got here some examples here of point mutations. This one here is looking at sickle cell anemia, and what happens on it happens on chromosome 11, and what happens is that here you've got GAG, and what happens is you get this substitution that happens here where A is substituted with T, and that results in an amino acid called glutamate being replaced with an amino acid called valine. Now you don't need to know this information here, it's just to give you a real life example of what happens, but because of that substitution it has a vast effect on the haemoglobin molecule, and it makes the haemoglobin, it links with other molecules, and that makes a very stiff molecule, and that makes the red blood cell bend, hence the reason why it's called sickle cell, because it changes into a sort of sickle cell shape. And that can have very, very serious consequences to the individual, depending on whether they've inherited both of these mutations. And you can go onto this website here and have a wee look at the information from that. You'll need to stop the video and copy this link, or type that link in. The hyperlinks won't work on the video. So that was our gene mutations. We're going to go on and have a look at splice site mutations. Now remember that you've got RNA splicing, which creates your mature mRNA transcript. Now if you've got mutations in the areas that mark the start or the end of an intron, then what happens is you can have one or more introns not being removed, and that will make a protein that doesn't function properly. To have a wee look at what I'm talking about here, there is an example here of a disease called thalassemia. You can always look that up. It's also caused it's caused by a defect in haemoglobin synthesis, and it's caused by a splice site mutation. Now what happens is, you'll see over here we've got normal splicing, and the introns are spliced out nicely, and you end up with a mature transcript, and you get a normal haemoglobin molecule that's formed. With thalassemia though, what happens is the splicing goes wrong, and you'll see here that there's an intron that has been left in when it shouldn't be, and that what happens there is that haemoglobin that is formed only has 2 oxygen carrying subunits instead of 4, and that has quite serious consequences for individuals, and you can look that up as well. So that is your gene mutations and your splice site mutations, and in the next video we'll move on to look at chromosome mutations and polyploidy.